that you know about prokaryotes from the last study guide, we will now take a look at its counterpart, eukaryotes. In eukaryotes, its cell organization involves a lot of packaging, and it is vastly more complex than that of prokaryotes. Their complexity allows eukaryotes to do things like carry out the metabolic reactions required to build a multicellular organism with cells that serve different functions. This is achieved by specialized cellular machines, called organelles, that are located within the cell membrane of eukaryotes. Let's take a look at some of these organelles. The nucleus of the cell is the largest and one of the defining features of eukaryotes. The nucleus is located in the center of the cell, and its nuclear envelope keeps it separate from the cytoplasm and other organelles, providing secure housing for the cell's genetic material. DNA in eukaryotes is organized into linear rather than circular molecules, as it is in prokaryotes. The nucleus is the site of DNA replication and RNA synthesis. Because RNA and other molecules need to travel between the nucleus and the cell's cytoplasm, the nuclear membrane contains little pores that allow select molecules' passage in and out of the nucleus. The nucleolus is a spherical structure within the nucleus, in charge of making and assembling the large and small subunits of ribosomes and transcribing the genes of ribosomal RNA, or rRNA. The nucleolus does not contain chromosomes, but does contain a gene to encode rRNA. In addition, they are responsible for moving ribosomes and ribosomal RNAs out of the nucleus and into the cytoplasm, where synthesis of the ribosomal proteins occurs. The endoplasmic reticulum, or the ER, acts partly as a highway for proteins and other molecules to move through, and also as a factory for the assembly of proteins and lipids that are needed for the function of the cell. The two types of ER, smooth and rough, appear to be separate when viewed under a microscope, but are in fact different compartments of the same organelle. The membranes of the rough ER appear to be a network of flat, sealed sacs, with the outside being bumpy by the presence of small, round organelles called ribosomes. These are molecules made in the nucleus that synthesize proteins out of amino acids and polypeptide chains. The rough ER provides quality control for these proteins and organizes them to be sent to their final destination. Unlike the rough ER, the smooth ER lacks protein-making ribosomes and appears more tubular and flat. It is also tasked with making and packaging molecules, but they make lipids and steroid hormones instead. Not only that, it plays a part in detoxifying by converting organic chemicals into water-soluble molecules. In the presence of an abundance of toxins, the smooth ER can grow to double its surface area in a short amount of time and return to normal size when toxins are no longer present in large quantities. The Golgi apparatus works like a post office because it is responsible for receiving the proteins and lipids from the rough ER, and to continue processing them until they're ready to be packed into sealed droplets called vesicles. These vesicles leave the Golgi apparatus bound for a variety of locations, inside or outside the cell. One of these vesicles are called lysosomes, which are responsible for various processes. Inside lysosomes are hydrolytic digestive enzymes that are used to break down macromolecules like proteins and carbohydrates into smaller molecules like amino acids and simple sugars, and to break down viruses and bacteria that invade the cell. They also break down organelles that are no longer working through a process called autophagy and consume excess products secreted by the cell in a process called chronophagy. A peroxisome is also an organelle that breaks down organic molecules like lipid chains, uric acid, and amino acids. They also protect the cell from the damaging effects of hydrogen peroxide, which is a byproduct of certain enzymes in the cell. Using an enzyme called catalase, The peroxisome isolates hydrogen peroxide and breaks it down into water and oxygen. Vacuoles are organelles that store and get rid of waste products, and also to maintain the shape of the cell. They store water and anything the cell has in excess, like waste products. Mitochondria are organelles that generate chemical energy used to run the cell's biochemical reactions. They do this by making adenosine triphosphate, or ATP, which stores and carries energy around the cell. The triple phosphate in ATP is a high-energy bond that will release the energy to fuel cellular processes by splitting off one of its 3 phosphates. Now that we've gone through organelles that are found in all eukaryotes, let's take a look at some organelles that are exclusive to plant cells. Chloroplasts are disc-shaped organelles with a triple membrane structure. In photosynthesis, chloroplasts turn carbon dioxide into sugars and oxygen. Chloroplasts are full of green, chlorophyll-containing membrane discs called thylakoids, the site of electron transport and ATP synthesis, which are often arranged into stacks called grana. In plant cells, mitochondria and chloroplast work together to make ATP from energy that is made from sunlight. Chromoplasts and leucoplasts are also plastid organelles found in plants. Chromoplasts contain carotenoids that are responsible for the yellow, orange, and red hue flowers, fruits, and other plant parts. Leucoplasts don't contain pigment, but they store starch and oil in non-photosynthetic tissues. Now that you know the parts of eukaryotes, you know a little bit more about how some organisms function, including yourself. That's everything we have in this lesson. If you need to review what you learned, go to www.scienceaide.com and access our study guide, download the PDF, and learn science smarter.